Hello, 8th standard CBSE children. Have a good day. Have a nice day. I hope you are doing well. Today we are going to do video number 18 as chapter 8 cell structure and functions, continuation of yesterday's lesson as refresher 2. I use the word refresher because it can be summing up of the lesson, discussion of the lesson, a mini teaching of the lesson, includes everything. So I have given a refresher. Okay, this lesson we already learned. I am recollecting the points. Right? Now this cell structure, yesterday we have learned it as structural and functional unit of life. Robert Hooke first identified the presence of cells in the cork, which is looking like a honeycomb cells. Robert Hooke in the year 1665. Yeah. Now this further studies, further invention, further advancement of microscope, other parts of the cells have been identified. As first, Robert Hooke in the year 1665 and coined the term cell and discovered this honeycomb-like compartment in a cork cell. Okay. Then it was Anton von Leeuwenhoek in the year 1674 studied the structure of bacteria, protozoan under the simple microscope. In our biolab we are having that is called compound microscope. And certain invisible items can be seen under this microscope. Right? Then Robert Brown was the scientist who coined the term nucleus. So, first, Robert Hooke identified the presence of cells in a honeycomb-like compartment in the cork cell. And then Anton von Leeuwenhoek studied the structure of bacteria and protozoa. And Robert Brown coined the term protoplasm. Robert Brown discovered the structure of nucleus. And it was Vacuole who coined the term as protoplasm. What do we know? Yesterday we have done. Yeah. Now this protoplasm, as we know, protoplasm, it is as nucleus and cytoplasm. It is as nucleus and cytoplasm. In yesterday's video session we have seen. Yeah? So this nucleus and cytoplasm together called protoplasm. And it is coined by the scientist Vacuole. Right? And yesterday we have studied the structure of nucleus. We will recollect. And then is the cytoplasm that includes living and non-living inclusions. Living inclusions are cell organelles. What I said? Cell organelles. Organelles now, the membrane bound structures found within the cytoplasm that is known as organelle. The membrane bound structure found within the cytoplasm that is known as organelle. Right? First inclusion, cell organelle, it is mitochondria. Cell organelle, this is mitochondria, where it is having two membrane, outer and inner. And outer membrane is smooth in nature, and inner membrane it is thrown into fold like this and called cristae. This is called cristae. Crista singular, cristae plural. and this is outer membrane. I hope you can understand this. Yeah? So what I said, நம்ம புக்ல வந்து கொஞ்சம் கொஞ்சம் தான் கொடுத்துருக்கு இந்த செல் இன்க்ளூஷன்ஸ் சைட்டோபிளாசத்தில். ஆனால் நம்ம வந்து கொஞ்சம் டீட்டெயில்டாக அதோட ஸ்ட்ரக்சரை படிக்கணும். சரியா? ஸோ ஃபர்ஸ்ட் வாட் வி ஆர் லேர்னிங் இஸ் மைட்டோகாண்ட்ரியா. What I said, mitochondria all living cells receive their supply of energy with the help of mitochondria. I repeat, all living cells receive their supply of energy with the help of mitochondria. Right? They are very often referred as powerhouse of the cell, and they are cylindrical in shape. Otherwise, we say they are more or less oval in shape, bound by two membranes, outer and inner. Outer membrane is smooth in nature, and inner membrane is thrown into folds called crista. Crista singular, cristae plural. Right? And these folds increase the surface area. These folds increase the surface area that can generate the energy rich substance. These folds, inner folds, increase the surface area that can generate the energy rich substance that is called ATP. Energy rich substance that is called ATP as adenosine triphosphate. ATP adenosine triphosphate. Sure? And then this cristae, it is having a pin-headed body. The cristalla is having a pin-headed body like this in every fold cell, and this is called F1 particle. Only this F1 particle region is generating energy, right? So F1 particle, it is also called oxysome. Just think F1 particle easier remember panna. And it play important role in cell respiration. During respiration, energy is released. So that is taking place during F1 particle releases energy. 1 minute. Okay. So this F1 particle, it can generate energy. I hope you understand this. Then the area inside the inner matrix, the space area, that is called matrix. I repeat. Now we say cell is having nucleus and cytoplasm. Now this cytoplasm, it gets different name in different places of the cell. For example, in mitochondria, it is within the cell organelle. So that cytoplasm, it is referred to as matrix. Namangada cytoplasm sollamattom. Same way, nucleus, within the nucleus, it is actually the fluid part, the cytoplasm sollapradhu. It is known as nucleoplasm. So like that, the matrix gets different names. The matrix contains enzymes that produce molecules used in the ATP. So functions of mitochondria: synthesize energy-rich compound ATP, adenosine triphosphate. Mitochondria synthesize energy-rich compound ATP. Okay, and the mitochondrial matrix also contains DNA and ribosomes. It is called mitochondrial DNA and ribosome. So as it has a little amount of DNA, deoxyribonucleic acid, it can synthesize its own cell. So it is also known as semi-autonomous body. Okay, so this mitochondria, as it contains little amount of DNA. It is also known as semi-autonomous body. Right? Then this presence of mitochondrial DNA and ribosome, it makes the cell as the mitochondria as a unique organelle. I hope you understand. The structure of mitochondria நம்ம bookல detailed இல்லைனாலும் நான் QBல தந்திருக்கேன். அதையும் வச்சுட்டு இதையும் you can write in your own sentences. I hope you understand this. Then second one, it is Golgi apparatus. Second one, it is Golgi apparatus. This Golgi apparatus, it is also called Golgi complex, and in plant cell, it is known as dictyosome. This plant, in plant cell, the Golgi apparatus, which is also known as Golgi complex, it is called dictyosome. Now, what is the structure of Golgi complex? It is formed of this Golgi complex. It is formed of flattened disc like this. There are many flattened discs like this, and then the vesicles, globular vesicles. Okay, so this is flattened disc. This is called cisternae, and this is vesicle. Okay, so now this the flattened structure, as I said, see here the diagram. நான் ஏற்கனவே நமக்கு previous videosல காமிச்சிருக்கேன், recollect பண்ணிக்கோ. Now this flattened structure, it is called cisternae, and these vesicles are the globular structures. They are called vesicles. I hope you understand. So now this Golgi body, it is discovered by the scientist Camillo Golgi. The Golgi apparatus, it is discovered by Camillo Golgi, the scientist Camillo Golgi. And then this Golgi complex, it is a collection of Golgi body with the flattened sac-like compartment that is arranged in stacks. Stacks na one above the other. And this apparatus was first discovered by Camillo Golgi. Right? They work closely with the endoplasmic reticulum. They work closely with endoplasmic reticulum, and they form a package in the form of vesicles or cisternae. Vesicle na globular structure, cisternae na flattened structure, and transport nutrients to various parts of the cell. So this is the structure of Golgi complex. Okay. Now what is the function? Golgi apparatus, it is involved in the formation of lysosome. Golgi apparatus, it is involved in the formation of lysosome. It is also responsible for synthesis of cell wall and cell membrane. It is also responsible for synthesis of cell wall and cell membrane. I hope you understand this. So this is for the structure of Golgi apparatus. Refer to these two figures, mitochondria and Golgi apparatus. Go through panavaranusha. Spelling padi. Camillo Golgi. Read the spelling. Adenosine triphosphate. Go through the spelling. Stay spelling for Okay. The next one, it is endoplasmic reticulum. Next one, it is endoplasmic reticulum. As this is the plant cell structure. Okay, this is a plant cell structure. Now this plant cell is having nucleus, and large vacuole. Nucleus and large vacuole. Okay. Now this endoplasmic reticulum, actually it is a continuation transport channel from the nucleus to the plasma membrane. Yesterday we studied about the cell wall, cell membrane. Plasma membrane, plasma lemma or cell membrane. Yeah. So it is having structure like this. So the nucleus layer, this endoplasmic reticulum is in continuation from the outer cell membrane to the nuclear membrane. Outer layer of the nuclear membrane. So now this endoplasmic reticulum is having dot like this on the outer layer of the membrane, outer layer of the reticulum. So now this endoplasmic reticulum, one minute, this endoplasmic reticulum, it is of two types. One is smooth endoplasmic reticulum, another one is rough endoplasmic reticulum. Now the smooth endoplasmic reticulum is, as the name implies, smooth in nature. Whereas this rough endoplasmic reticulum is having these blue dots, ribosome. Rough endoplasmic reticulum is having ribosomes. These blue dots, they are called ribosome. So the endoplasmic reticulum which is having ribosome, they are called rough endoplasmic reticulum. When it is not necessary that all the transport of the network of ER will have ribosome. So now endoplasmic reticulum, it is an interconnecting system of channels. Endoplasmic reticulum is a interconnecting system of channel and tubules that looks like sacs and fold. Endoplasmic reticulum is an interconnecting system of channels and tubules that look like sacs and folds. It is spread throughout the cytoplasm and is in continuation with the plasma membrane and nuclear membrane. The plasma membrane, the outer the nuclear membrane. Okay. Now there are two types of endoplasmic reticulum. Another in short ER chelluva. So it is smooth and rough. Smooth endoplasmic reticulum SER, rough RER. Right? The smooth endoplasmic reticulum are found in cells which synthesize steroids, hormone and lipids. What is the function? Synthesizing steroids, hormones and lipids. Okay. So this smooth endoplasmic reticulum synthesize steroids, hormones and lipids. Okay. Steroids, hormones and lipids. Whereas rough endoplasmic reticulum, it is taking part in protein synthesis. Rough endoplasmic reticulum, it is taking part in protein synthesis. I hope you understand this. So I repeat, the smooth endoplasmic reticulum are found in cells which synthesize steroids, hormones and lipids. The walls are smooth and form tubules. Okay, the rough endoplasmic reticulum, they are synthesizing protein. This type of endoplasmic reticulum has bumpy looking walls. In the mari dot bumpy looking walls because of the attachment of ribosomes, and it is playing a very important role in protein synthesis. I hope you understand. So we are doing 3 cell organelles. First one, mitochondria. Second, Golgi body. And third one, it is endoplasmic reticulum. Are you clear? So third one is endoplasmic reticulum. Right? Then fourth one is vacuole. Vacuole enna sollirukken? This is the vacuole. Now in this vacuole, in plant cell, its presence is very important characteristic feature. Actually, plant cell can be differentiated from the animal cell by the presence of definite cell wall giving definite shape, and then presence of large vacuole, and third one, presence of plastids, right? Whereas these 3 are absent in animal cell. I hope you understand. So now this lining layer of vacuole, it is called tonoplast. This lining layer of vacuole is called tonoplast. And then it encloses, it is not empty, it encloses cell sap, the fluid nutrient contains, or cell sap. So large fluid filled sac called vacuoles are found more in plant cells than in animal cells. Mature plant cells are found to have one large vacuole that almost fills up the entire cell. They play important part as contracted vacuole, where excess water and waste are excreted from the cell. Now in unicellular organism amoeba, this vacuole plays an important role as when it digests the food, further digestion, it is called food vacuole, and then when it is taking excretion process and maintaining the water balance, it is as osmoregulatory in function, and that vacuole is called contractile vacuole. I hope you understand this. They also function as food vacuole, where they engulf food material. Example, amoeba, the food vacuole engulfs food item and digesting it using digestive juices. I hope you understand this. So this is vacuole. The function, it stores and concentrates mineral salts and also nutrients. Vacuole stores and concentrate. Vacuoles store concentrate mineral salts as well as nutrients, and it maintains proper osmotic pressure in the cell to maintain its rigidity. So it maintains osmotic pressure in the cell to maintain rigidity. I hope you follow this. So this is vacuole, and in our textbook this vacuole is giving the especially in book example onion peel. So the blank looking structure, this is blank looking structure, but it is not empty. It is not blank looking. நம்ம பார்க்குறது அப்படி இருக்கும். But it contains the cell sap. The blank looking cell is called vacuole. Okay, we'll just continue. This vacuole, as I said earlier, the outer covering, it is called outer lining layer is known as tonoplast. சரி, நம்ம bookல இல்லை, ஆனா note பண்ணிக்கோ. Okay, so now this vacuole, it is present in large size in plant cell, whereas in cheek cell, animal cell, it is smaller. Normally, vacuoles are absent in animal cell, and if at all it is present, it is smaller in size. I hope you follow this. So large vacuoles are common in plant cell, and it is absent, but if present, it is very much smaller in animal cells. So that is for the vacuole structure. Right? The next one, it is plastid. Now this plastid, actually it is of three types. Plastids is of three types: chloroplast, chromoplast, chromoplast and leucoplast. There are three types of plastids as chloroplast, chromoplast and leucoplast. As we know, chloroplast, we are studying here. It contains the green pigment chlorophyll, and it is most abundant in green leaves. Green plants having chloroplast, which contains the pigment chlorophyll. Whereas this chromoplast, it is having different coloration, and that colored pigment as xanthophyll, anthocyanin, these pigment, they are present in flowers, different colored flowers we have seen here, those colors, and in fruits also it gives the coloration, especially the pigment xanthophyll and anthocyanin, and then carotene in carrot. These are the chromoplast pigments. Whereas this leucoplast, it is colorless. Normally we say the colorless plastids are called leucoplast. What does it mean? Typical example, apple சொல்றோம்ல, apple fruit we know. That apple, the fleshy portion that we are using, that we are eating, அது என்ன கலர் சொல்லுவ? ஒயிட்டும் சொல்ல முடியாது, ப்ரௌனும் சொல்ல முடியாது, எல்லோனும் சொல்ல முடியாது இல்லையா? ஸோ இட் இஸ் கலர்லெஸ் பிக்மெண்ட். பட் கலர்லெஸ் பிக்மெண்ட்னா அது ஒன்றும் பிளாக்காக இருக்காது, ஏதாவது ஒரு லைட் டிஞ்சு கொடுக்கும். ஸோ தட் கலர்லெஸ் plastids, they are called leucoplasts. So I repeat, plastids are of three types: chloroplast, chromoplast and leucoplast. சரியா? நம்ம புக்ல chloroplastோட structure கொடுத்திருக்கு. Again, it is more or less oval or elliptical in nature as outer and inner layer. More or less oval or elliptical in nature as outer and inner layer. சரியா? And inner to that is the matrix. Cytoplasm சொல்ல மாட்டோம் இல்லையா? So this is called matrix, right? Now this matrix, it is stroma. This matrix in chloroplast, it is stroma, right? And this stroma, they are found inside the chloroplast as lamellar structure like this. Lamellar structure like this. So this is called stroma lamellae. So this is called stroma lamellae. Now this stroma lamellae, they are also formed of chlorophyll pigments, right? And these chlorophyll pigments, they are concentrating in patches like this. Concentrated chlorophyll pigments are found as patches like this. And these green patches are called grana. Grana singular, grana plural. And this is the site for photosynthesis. Chlorophyll pigment, they are much concentrated in this granum region. Okay. Now this granum, when they are piled up like a coin, like this, piled up like a coin, என்னன்னா நம்ம ஒரு 10 1 rupee coin அப்படி அடுக்கி வச்சோம்னா இந்த வளைவு வளைவா தெரியும்ல, அதான் piled up like a coin or stack of coin. So these grana, when they are coiled like a, when they are packed like a coin, they are called thylakoid. That concentrated region, it is called thylakoid. So actually during photosynthesis, when the sunlight falls on the leaf, the active site for photosynthesis is the thylakoid, where the chlorophyll pigment, it is much concentrated. I hope you follow this. So in our textbook, this the chloroplasts, they are scattered in the cytoplasm. In a textbook like example கொடுத்துக்கிறது leaf வந்து Tradescantia leaf னு சொல்லிருப்பாங்க. Tradescantia leaf. And they are different colors. Plastids are of different colors. Some of them are green pigment having chlorophyll. Green colored plastids, they are called chloroplasts. And they provide green color to the leaf. Now this chloroplast again one minute. This chloroplast, then chromoplast and leucoplast படிச்சோம். The colored pigments, chloroplast green color, chromoplast வந்து different colored pigment, and leucoplasts are colorless and help to store starch, oil and protein molecules. Leucoplast, they are colorless plastic and help to store starch, oil and protein molecules. Now this chloroplast, as I said, will be having structure like this. நம்ம முன்னாடி வீடியோல காமிச்சிருக்கேன், ஞாபகப்படுத்திக்கோ. Structure like this. எதெல்லாம்? Phylum, coil, granum, the cytoplasmic matrix, the matrix பேரு stroma, that contain stroma lamellae. Right? When the granum, when they are piled up like a coin, it is called thylakoid. I hope you understand. So each chloroplast consists of double-membered membrane envelope and a matrix. The inner membrane is arranged along the length of the plastids as lamellae. At certain region, the lamellae are thickened and appear like pile of coins. These are grana, and each grana having a disc-shaped membranous sac. And the photosynthetic pigment chlorophyll, it is located in the thylakoid. The non-thylakoid part of the matrix is called stroma, and it contains number of enzymes involved in photosynthesis. I hope you understand this. So this is for the structure of chloroplast, right? And then centrosome, it is present in animal cell and in certain lower animals. And normally it is absent in prokaryotic cell and present in higher plant cell. And the centrosome is located in the cytoplasm just outside the nucleus and contains a pair of small hollow granules called centrioles. So this is the structure of centrosome. I hope you understand this. So now in our textbook, it is given as, one minute, in our textbook for cell structure and function. First, cell is the structural and functional unit. Discovery of the cell. Robert Hooke in the year 1665 identified the honeycomb-like compartment cells in the cork. And then Robert Brown discovered nucleus. Anton von Leeuwenhoek discovered the structure of bacteria and certain protozoans. Pickenji coined the term protoplasm, which is formed of nucleus and cytoplasm. So this is the discovery of the cell. And then about the cell, as we know, it is the structural and functional unit. Then cell size and shape especially size and shape. It is amoeboid in amoeba and WBC, and normally the cell with the single membrane doing all the physiological function, they are called prokaryotic cell, and all other cells, when they have a regularized different organ system, they are included under multicellular organism. Cell, tissue, organ systems and organism. Okay, and so this is unicellular and multicellular. Shape of the cell, amoeba is an organism showing movement by putting forth pseudopodia, whereas WBC, white blood cell, it is present in the blood tissue and it is a cell showing amoeboid movement. RBC, red blood corpuscle, it is a cell. Smooth muscle is a cell. Long branched neuron is a cell. Okay, and then a group of cells doing having similar structure and function, they are called tissues. That also you could recollect, right? And then parts of the cellular, cell membrane or plasma membrane, and then about the cell wall partition. I hope you remember this. Then in the cytoplasm, the cell organelles inniki paathonda adella. Mitochondria, Golgi body, Endoplasmic Reticulum, lysosome, ribosome, plastids, and centrosome. This lysosome, it is also known as suicidal bag, where it is digesting or engulfing all the destroyed and worn out tissues. And then we are learning about the structure of nucleus as prokaryotic and eukaryotic nucleus. I hope you remember this. And then as nucleus, it is having nucleus, nucleoplasm, nucleolus, chromatin network, that is chromosome. That chromosome contains chromatin matter matrix, which in turn having DNA, that DNA in turn having genes. I hope you remember that. So this is given in our textbook. Okay, that you can recollect. Now the difference between prokaryotic and eukaryotic cell. What do we know? First, prokaryotes, they are smaller in size, and it is lacking well-defined nucleus. Only the genetic material is represented, and that is called nucleoid or incipient nucleus. And then this prokaryotic cell lacks membrane bound cell organelles, and ribosomes are smaller in nature. Whereas eukaryotic cell, they are larger in size. They have well-defined nucleus and nucleus with more than one chromosome, and nucleolus, it is absent in prokaryotes, present in eukaryotic cell. And this eukaryotes, they are having well-formed membrane-bound organelles, and ribosomes are larger in size. And cell division, it is taking place as mitosis and meiosis. Okay, and then as this cell structure, இந்த diagram தான் நான் கொடுத்த இந்த previous videoல அதை மட்டும் கொஞ்சம் go through பண்ணிக்கோ. Structure of plant cell and animal cell. I hope you understand. And then difference between plant cell and animal cell. என்ன தெரியும்? Plant cell has outer rigid cell wall which is made up of cellulose. Note பண்ணிக்க, நம்ம bookல இருக்கு. Plant cell has outer rigid cell wall which is made up of cellulose. Plant cells are larger than animal cell. Plant cell has large vacuole which occupy more space of the cell. Very large vacuole. In that case, nucleus will be pushed towards one side. Right? Centrosome is present only in some lower plants. And lysosomes, again, they are found only in eukaryotic plant cells. And plant cell contains plastids. And starch is the storage material. So this is for the plant cell. Whereas animal cell, it lacks cell wall. Animal cell is comparatively smaller in size than the plant cell. It lacks vacuole. Even if present, as I said, they are very smaller in size. And all animal cells contain centrosome. And lysosomes are found in all animal cells. And plastids are absent. Glycogen is the storage material. I hope you understand this. So this is for the cell structure and function. Okay. In our textbook, it is a cell structure and function. Okay. Now this chromosome, one more just I will say. Now this chromosome, as I said, it is having a centromere and two arms. Now these arms, it is called a pellicle. Okay. Inside that, it contains chromatic it contains chromatic matter. Okay. That in turn having chromosome, that in turn containing DNA, chromatic matter, which in turn having DNA, deoxyribonucleic acid, and that in turn contains genes. I hope you follow this. Now this chromosome, these two arms, depending on the size and position of the centromere. For example, take this. These two arms, they are equal in nature. So it is called metacentric chromosome. Chromosomes with two equal arms, they are called metacentric chromosome. And with one arm shorter and another arm larger, it is longer, it is called submetacentric chromosome. Okay. The two arms with one arm smaller and another longer, it is called submetacentric chromosome. And this chromosome is called actocentric, and one arm is very short and another arm is normal longer size. that is acrocentric. And it is telocentric, where one arm is totally absent and only it is having single arm. That is called telocentric chromosome. I hope you understand. So now, based on the position of the centromere, the chromosomes can be metacentric, submetacentric, acrocentric and telocentric chromosome. I hope you understand this. And then about the structure of DNA churikilla. Another DNA, it is deoxyribosugar, and it is in double helix or double spiral, and it is proposed by the scientists Watson and Crick. So it is called Watson and Crick model, right? So chromosomes are made up of a long series of structures called genes. Genes are made of chemical called deoxyribonucleic acid, and each DNA strand is made up of millions of nucleotides, and each nucleotide is made up of a pentose sugar, phosphate group and nitrogenous base. Okay. But the nitrogenous bases are two kinds, as I said earlier. It is purine and pyrimidine. Purines are adenine and guanine, and pyrimidines are thymine and cytosine. And these purine and pyrimidine, they are always located in the DNA in pair, and it is called Chargaff's law, as adenine will go with the thymine and guanine will go with cytosine. That's why ATCG chalo. And then the structure of DNA proposed by Watson and Crick. DNA is a double-stranded helix formed of deoxyribosugar. And actually the backbone of helix, it is formed of sugar and phosphate molecules. The nitrogenous bases are attached to the sugar molecules. I hope you understand. The two polynucleotide strands are held together by hydrogen bond between specific pairs of purine and pyrimidine. Specific pair naka adenine will go with thymine, guanine will go with cytosine. I hope you understand this. So this is for the cell structure and function. Okay. The eukaryotic a eukaryotic cell has well organized nucleus. It also has structures like endoplasmic reticulum, Golgi body, mitochondria, plastids and vacuoles. Normally, in plant cell, presence of, as I said earlier, presence of cell wall, plastids and vacuole are the characteristic features, right? And these specialized structures, they are found in cell called cell organelle. I repeat, the membrane bound structure present inside the cytoplasm, they are called cell organelle. Okay. Genetic material or chromosome is also found enclosed in a membrane bound structure called nucleus. And protozoan, unicellular algae and fungi have eukaryotic cells, and all plants and animals have eukaryotic cell. Protozoan, unicellular algae and bacteria, they are unicellular, and all other plants and higher animals, they are as eukaryotic cell. சரியா? So இந்த diagram என்ன ஒரு பாரு, நான் ஏற்கனவே கொடுத்திருக்கேன். நம்ம QB ல இருக்கும். We will just refer. I hope you understand. சரியா? Yes. Then நம்ம புக்கில் 8.1 இருக்கு பாரு. இதில் கம்பேரிசன் ஆஃப் பிளான்ட் செல் அண்ட் அனிமல் செல் இருக்கு. செல் மெம்பரேன் போத் பிளான்ட் செல் அண்ட் அனிமல் செல் present. Cell wall plant cell present, absent in animal cell. Nucleus present in both plant cell and animal cell. Nuclear membrane present both in plant cell and animal cell. Cytoplasm present in both plant cell and animal cell. And plastids present in plant cell, absent in animal cell. And vacuole present in plant cell and absent in animal cell. I hope you understand this. Yes, sir. Right. And then this is human cheek cell. என்ன ஞாபகம் உனக்கு? Cheek cell. Vacuoles are smaller in size. In human cheek cell, vacuoles are present. They are smaller in size. Cheek cell இந்த இடம் தான் cheek. ஃபேஸில் இந்த இடத்துல கண்ணான்னு சொல்லுவோம் இல்லையா, அதுதான் cheek. ஓகே, ஸோ வேக்யூலை பொறுத்தளவில் it is larger in size in plant cell, absent in animal cell. If present, they are smaller in size. Presence of vacuole in animal cell வந்து நீ human cheek cell சொல்லி இந்த diagram வரையலாம். இந்த diagram you have to draw. I hope you understand. Are you clear? So this is for the cell structure and function. The diagram we will go through panel. Page 98 நம்ம bookல page 98 இருக்கு. அதையும் page 97 gene இருக்கு பாரு. Gene is a unit of inheritance in living organisms. It controls the transfer of hereditary characteristics from parents to offspring. I hope you understand. Go through panel. Jimmy. Okay, sir. And then what do you understand? Remember for tonoclast is the outer layer of the vacuole, and thylakoid the granum which is concentrated much with the chlorophyll, and when they are piled up like a stack of coin, it is called thylakoid. And thylakoid is the active site for the photosynthesis. When light falls on the leaf, the first active center is the thylakoid. I hope you understand this. Okay. So this is the structure of cell structure and function. We have just as a refresher. Okay. We'll continue in the next video session. Thank you. Have a nice day.